also for being part of Cure as an advisor and a friend from the beginning. I think that a very much anticipated presentation, because we want to hear and understand a little bit more from your experience into using bacteriophages as a medicine. So we have seen that your title that it is a medicine, Jim, but not as we know it. And some of us at least understand the reference. So please go ahead, and we're looking forward to our discussion.
Okay, thank you for having me. Good afternoon. Yeah, just quick refresher. Phages are, of course, the viruses of the bacteria. You can see an E. coli bacterium in yellow. You can see some phages in green. And you can see some of them injecting their DNA into the bacterium. Of course, the colors are fictional because this is an electron microscope picture, which is black and white, and it was colored afterwards. So in phage therapy, we use the lytic cycle of the phages. Again, to refresh, we get a phage that absorbs to the bacterium, as you saw. Then it will inject its DNA. It can also be RNA, but most of the time it's DNA. So it's genome into the bacterium. Now you can compare this to those alien creatures you all know, which attach to a face and inject their genome. This genome hijacks the machinery of the bacterium and forces it to replicate not the bacterium, but the phage genomes. And then it also forces the machinery to produce phage proteins, the envelope and the tail fibers. And what we get is phages fully developed inside of the bacterium. Can be a handful, can be hundreds. And then the bacterium explodes. Again, in comparison with those aliens which come out of the human. And we got ourselves a self-replicating antimicrobiome. We got interested in phage therapy in 2003. Some of you might have seen the slide already. This was the beginning, and as always, beginnings are difficult. So when we want to do research in the military, we need to write a proposal, and we need to send it to the military academy. That year, 2003, there were 26 proposals submitted, and these are evaluated by 11 experts, experts between brackets. 11 got funded. Where were we? I have to admit, because otherwise I cannot go further. Yeah. So we were not funded. We got 8 out of 20. And one of the evaluators had written on the proposal that it was science fiction. Now, as you know, this is a therapy that almost has 100 years of existence, and still someone said it was science fiction. But everything is relative, because the proposal scoring 7 out of 20 was one about collecting postcards featuring military uniforms pre-Waterloo 15. Of course, since then, we have obtained some funding. And in those 17 years, we have developed an entire chain of phage therapy research in the hospital, and we have a research laboratory where we do isolation and characterization. We have clean room facility for production. There's a pharmacy, and there's also the clinic where phages can be applied. So what is special is that everything is under one roof. And of course, the foundation of this are international collaborations, and the most important one, of course, with the Eliava Institute. And you see here a picture of the Eliava Institute. Since 1923, they perform phage therapy research and also applications. We have a long-time collaboration with Nina Chanishvili, who was honored, I would say, in a recent profile paper in the Lancet Infectious Diseases. And of course, we also have two of Eliava Institute's former students and researchers who are coordinating phage therapy research in the military hospital. And these are Maya Merabishvili and Thea Ronti. So, as you also know, in the Western world and the EU and the US, phages have been classified as medicines. The problem is that they were classified as medicines without any special framework. And you can here see the answer of the European Commission when they were questioned on the framework for phage therapy. They just answered that the existing medicinal product framework is adequate for bacteriophage therapy, and that was the end. So as you all know, this means that phages need to be produced according to good manufacturing practice. Not only that, every phage is considered a medicinal product, so it needs to be tested in preclinical tests, but also in phase 1, 2, 3 randomized controlled trials. So RCTs, which is the holy grail of clinical trials in the EU, but also in the US. This is not impossible, but this requires a lot of time and a lot of money. Typically 5 to 10 years and millions of euros or dollars. And when we say large investments, we also say strong IP protection. Problem is that phage therapy is in the public domain since the 1920s. And also since the Myriad Genetics case, we also know that it's unlikely to patent products of nature, such as phages. This case is about the Myriad Genetics company who had patented two breast cancer genes, BRCA1 and 2, to be used in diagnostic tests. Now, these patents were attacked by the Association for Molecular Pathology, who wanted to have this test available for everyone and not only produced by this company. And so there was a trial and the Association for Molecular Pathology won. And the Supreme Court said that, in fact, when you use a natural gene or natural DNA sequence, there is no invention. So still, there are some companies that are trying to develop these phage medicinal products. And there have been also a handful of phage therapy RCTs, randomized controlled trials. Now, these RCTs evaluated defined phage products and most often a standalone therapy, so only phages were used. In fact, they were tested as if they were new antibiotics. And these trials up to now showed rather disappointing results with regard to efficacy. And this is in contrast with the historical phage therapy applications, for instance, in Georgia, and also with recent Western case studies. So why is that? Well, of course, there is a positive result publication bias, because most often, especially in those case studies, only the positive cases are published. And when you look at those cases, you will see that phages are always used as adjunctive therapy. So they will be combined with, for instance, antibiotics. Also, these phages are most of the time are selected to target the patient's bacterium. It's a personalized therapy. And sometimes phages are pre-adapted or trained or even engineered to be more effective. So that's why I tend to say that what has been investigated in RCTs up till today is not phage therapy, it's defined phage products. Are they a medicine? Well, according to the definition, when you claim a therapeutic effect, you have a medicine. So they are medicines, but they are not medicines as we know them. So the problem is that these defined phage products, they underappreciate a number of phage peculiarities, a number of advantages those phages have over antibiotics. Now, the younger attendees will probably ask themselves, who is Jim? Now, Jim is a fictional captain, Captain James T. Kirk of the Starship Enterprise. And during their space travel, they often encountered new, strange life with characteristics that they were not familiar with. Just an example.
Captain, doesn't even look real.
It's not life as we know or understand it. And it is obviously alive. It exists.
And it can bear up under full phaser power.
So the first peculiarity would be that phages are species or even strain specific. So you all know antibiotics, or most of the antibiotics have a large spectrum of activity, and they can be used often empirically without testing. Phages are different. They target one species and most often only a part of the species or even a few strains. So they are very specific. This has some advantages. One of them that they will not disturb the patient's commensal flora. The disadvantage is that you need to know which bacterium is infecting. And this can be sometimes problematic because often you don't have the time to find out which bacterium is causing the infection. Now, when we look at phage therapy requests that were submitted to the phage therapy centers in San Diego in the US and in Brussels, we can see that over a period of approximately 1 year, these requests involved 35 species in San Diego and 31 species in Brussels. Now, let's say that you would need 5 phages per species, and this is a very optimistic number, that we are talking about 150 phages you would need. Also, you have some geographical variations between San Diego and Brussels in the species and the strains, and you will also have variation in time. So this means hundreds of phages. And all these phages are medicine products and need to be developed in GMP and tested in RCTs. You can see that this might be problematic. Now, this is an example you probably know. This is the Fagoburn trial, one of these RCTs, which evaluated the safety and efficacy of phage cocktail against Pseudomonas aeruginosa, burn wound infections. And this cocktail consisted of 12 lytic anti-Pseudomonas aeruginosa phages. What is less known is that in an ancillary study, in retrospective study, it was shown that 1 in 3 strains isolated from patients before phage treatment were resistant to the phage product. So even when using 12 phages, or 1 in 3 of the strains were resistant before therapy. Peculiarity 2, bacteria and phage are involved in host-parasite relationship. It's very complicated, but the bottom line is that phages will not eradicate their hosts. They are not stupid, they will not commit suicide. So it will be very difficult to eradicate bacteria using phages. We will need to count on the immune system of the patient or other antimicrobials. Of course, not to be eradicated, this means that bacteria need to quickly become resistant to phages. But the advantage is, of course, that new phages can emerge to again infect these resistant bacteria. So it's an arms race. It's a co-evolution between phage and bacteria. And you can imagine if you freeze phages in time, let's say you will only develop every 10 years a phage product, then you lose, of course, this co-evolving advantage. The advantage of this co-evolution is that you can train phages to become more active. So you select a good phage from collection, and then you can even match it even further to the infecting bacteria. That, of course, when you have time. There are in vitro studies, but also in some animal models that show that when you adapt the phage to the patient's bacterium, these phages are more efficient in clearing the pathogen, but also they will elicit less resistance of the bacteria. And this Friday, we will have a meeting on phage training in Phage Club, which is a club we created in a popular app, Clubhouse. And it's this Friday at 8 o'clock Central European time. An example of the emergence of resistance in vivo, not in vitro, but in vivo. And here, this is a 1-year-old patient with a Pseudomonas aeruginosa sepsis after post-liver transplant. And in the beginning, the bacterium was susceptible to the phages, but after a while, we started isolating we started isolating bacteria that were insensitive or less sensitive to these phages. And you can see here the genomes. The green ones are the sensitive bacteria, and then the red ones are resistant or less sensitive bacteria. And you can see quite some mutations. Interestingly is that these phage-insensitive mutants showed no reduced virulence in the Galleria melonella model. Another example is Pseudomonas aeruginosa contamination using a nasal phage spray. And also there, some phage-insensitive mutants were isolated. These mutants showed some reduced virulence in Galleria melonella. Not spectacular, but there was some reduced resistance. Now, importantly, when we took the same phages and the same bacteria and we evolved resistance in vitro, and we compared the in vitro mutants, resistant mutants, with the in vivo resistant mutants, we saw quite some differences. I don't have time to go into details, but there will be a manuscript out soon, I hope. But one of the important differences was that in vitro, the resistance to Pseudomonas was most often due to biofilm formation, while the in vivo resistant mutants showed no increased biofilm formation. So there were differences in resistance mechanisms, and also from genomic analysis we can see differences. Now, an example of phage training. We had a patient with Staphylococcus epidermidis infection. Then there's this very good phage from the Eliav Institute, phage ISP, which targets mainly Staphylococcus aureus, but it can also target some strains of Staphylococcus epidermidis. So it was tested against the patient's strain, and there was some activity, but it was not very stable activity. So Maya Mirabishvili then trained phage ISP, and she did 8 rounds of coevolution using Appelman's method on each of 3 clinical Staph epidermidis strains. And then she managed to have a stable activity for 48 hours without emergence of resistance at low multiplicity of infection. Now, when we looked at what this meant on the genome level, we saw 4 missense mutations. And when we looked at the host range, while phage ISP showed some low activity on 3 Staph epidermidis strains out of, say, 12, I think. We saw that the newly trained Staph epidermidis phage only targeted the strain of the patient. So it had become specialized, but it had lost host range. So it had been personalized to target the patient's strain. This brings us to the personalized approach, and this is the approach that we feel is most efficient when there is time, of course. And then you would start with a collection of phages, and you have your patient, you isolate the patient's infecting bacterial strain, you send it to the lab, and then you will select 1 or more good phages from your bank. If you have time, you will train them. If you cannot find phages in your bank, then you go and look in the environment. And then this phage is mixed. These phages are mixed, and they can be put into a carrier, which can, for instance, be a hydrogel or a buffer for IV injection, and then given to the patient. This personalized approach is, of course, more sustainable. You will of course get less resistance, for instance, compared to using large spectrum phage cocktails. You can compare it with what happened with antibiotics. And this is very much in line with what today is called evolutionary or Darwinian medicine concepts, where you will use the natural controls of bacterial populations to control infections. And infections are just like unrestrained bacteria. So that's what phages do in the environment. They will restrain uncontrolled bacteria, and you want them to do the same thing in patients. But of course, this is not compatible with the current practice, where, of course, these phages are wanted immediately. That's why we say that we should probably have both systems. So we should have those broad spectrum cocktails, as broad spectrum as possible, to add to antibiotics in first-line treatments where you don't have time. But when you have time, you should really go for the personalized approach. And of course, both systems are also complementary, because the good phages you will find in this personalized approach can go and be part of these phage cocktails. Now, after 2 decades of phage therapy renaissance, as it is called, and you can see this quite famous paper in Nature Biotechnology from 2004, where they were talking about a lot of companies that were moving into phage therapy. Well, we are now 17 years later, and we don't have any phage products on the market. And of note, until the 1990s, Institut Pasteur in France delivered phage preparations to pharmacies. You possibly know these high-profile phage therapy cases that have gotten some attention in the media. Of course, these put pressure on medical doctors to apply phage therapy in desperate cases. So we wanted a pragmatic solution for phage therapy. And in Belgium, what we did some lobbying, and you see here Dr. Maggie de Blok, our former Minister of Public Health, in Belgian Parliament in 2015, where she was confronted with questions from the opposition, the Green Party and the Socialist Party, on the status of phage therapy in Belgium. And there she answered that she was well aware of the problems of bringing phages to the market as industrially produced medicinal products. And importantly, she opened the door for what is called magistral preparations. And then also, very important, she tasked her authorities for medicines to help us to set up a more pragmatic framework. What are magistral preparations? Well, they are part of European and Belgian law. Magistral preparation is defined as any medicinal product prepared in a pharmacy in accordance with a medical prescription, so personalized, for an individual patient. So this framework is a problem, of course, when you want to develop broad spectrum cocktails, but this is very much in line with the personalized approach, where you will have a medical doctor prescribing phages for an individual patient. Now, the active pharmaceutical ingredients, or APIs, of magistral preparations, in this case, are the phages. And these phages must conform to the provisions of a monograph. And a monograph is like a recipe. It describes how phages need to be produced and tested. It also describes quality testing. And the document you see here describes how to produce phages and also to test them. What is the concept? So we have a phage bank with seed lots. These are characterized phages, and they are stored using a banking system. So we can train them, we can evolve them, but once in a while you need to be able to go back to the initial phage. You cannot have unlimited drifting of your phage, because after a while you will get a totally different phage. And this is not yet accepted in the system. To enter the phage bank, these phages need to be tested by an external approved laboratory. And this testing consists of a genome analysis, so sequencing of the entire gene. So sequencing of the entire genome is performed, and then some specialists, they screen this genome for unwanted genetic determinants, For instance, lysogeny-related genes, antibiotics resistant genes, toxins, etc. But also the production strain, so the bacterium that you will use to produce the phage, is also screened. So its genome is also screened for toxins, for instance. Quantitative PCR is validated, and this quantitative PCR will be used later to identify the correct phage in batch productions of this phage, and also to quantify the phage. This is the end of such a quality testing report. What is analyzed is the safety of the phage. In this case, the phage is approved and it can enter the phage bank. So once it's in the phage bank, it can be used as a seed lot to produce phage APIs, so phage active pharmaceutical ingredients. And you need to produce them according to the monograph, and also again send out some samples for quality testing. So I need to stress that this genomic map is performed once for a phage, only one time. And these quality tests that you see here, they are performed for every batch that is produced from these phages that are in the bank. Now, and this testing is very pragmatic. It's endotoxin content, it's bioburden, and its pH. Nothing more than that. So very pragmatic. Also, you see here, this phage is approved. So this batch has been approved for use in magistral preparations. So I'm always asked, so your phages are GMP certified? No, they are not GMP certified. You can if you want, but there's no need for. This does not mean that there are no standard operating procedures, or there are no there's no control of the production environment. Not at all, but there's no formal GMP certification, and it's that part that costs a lot of money. So, as I said, these phages can be used in magistral preparations. They go to the hospital pharmacy together with the quality testing certificates. And there, upon prescription by an MD, they can be integrated in phage magistral preparations. So you can mix different phages in, for instance, an hydrogel for burn wounds, or in an IV solution to treat sepsis. These are examples of these magistral phage preparations. Of course, when you send phage APIs to the pharmacy, the pharmacist wants to know what is the stability of these products. How do I need to store them, and how long can I store them? So we did a lot of tests on the phages in different buffers and different creams. And there are huge differences. You see here, for instance, when you mix phages with 13 creams that are used in burn wound treatment, you can see that in some creams, the activity of phages goes down very quickly, at least of some phages. It's also phage dependent. While in other creams, the activity stays stable for longer periods. So you see in this cream already after 30 minutes, the activity is almost totally gone, while in other creams, 24 hours, the activity is almost 100%. So these are all things that medical doctors and pharmacists need to know. Also, for instance, when you will use phages in nebulizers, it's also very important to know which nebulizers. And this is an example of a jet nebulizer. And you see that some phages, will go down in activity with 2 logs, which is huge. Huh? So you keep 1% of the activity. But we see that when you use mesh nebulizers, you don't have that problem, or much less. Also, if you want to store your phages for long periods, it's better to dry them or to freeze dry them. And we have now we are partners in a big project drying and encapsulating phages. If you want to know all the details about this magistral phage framework, this has been published open access, and the phage monograph is supplementary material to this publication. We published this in collaboration with the Federal Agency for Medicines and Health Products and with the Belgian Institute for Health. So it was really a good collaboration between the hospital, but also the authorities. But to be clear, this is only possible when the politicians, they ask those people from the authorities to collaborate with us. So since 2018, we have this framework in Belgium. And because we had a lot of requests from other countries in Europe, we are now trying to put this phage monograph into the European Pharmacopoeia. And this is a lot of logistics and administrative work, which was very difficult during COVID-19 times. But there have been quite some meetings now, and the European Commission agrees in principle, and there is a dedicated committee which is discussing the introduction of the phage monograph into the European Pharmacopoeia. So the last part, of course, is you have to bring the phages to the patients and to have phage therapy. So we have coordinated treatments in more than 70 patients, which of course compared to the Yave Institute is nothing. But for a Western European country, this is something, I think. In more than 20 hospitals and more than 10 countries. And you see a sharp increase in the deliveries of phages. There was a oral presentation a few days ago at the ECMID conference by Sarah Jebara, who is coordinating phage therapy in the military hospital. And I think that ECMID members can see this presentation online. It has been recorded. I will give a very quick overview of these treatments. So 66 of these treatments have been analyzed. Of course, since a lot of them were performed in other hospitals and in other countries, it was often very difficult or even impossible to obtain data. Here you can see the indications for which the phages have been used. Most often lower respiratory tract infection and bone and prosthetic joint infection. Now the outcome. So we looked at microbial eradication, and you can see in some cases there was microbial eradication. We looked at clinical improvement. You also see some clinical improvement, but you can also see that some cases were unsuccessful. But we can see clearly that the cases where phage therapy was most often successful, where we had microbial eradication, were in bloodstream infection and in bone and prosthetic joint infection. What is our experience with this magistral phage framework? Well, we have more applications now by different MDs in different hospitals. problem is, of course, that they all have their own treatment protocols and also sampling protocols. They all want to publish their own cases. It would be nice if we would have like some kind of joint publication on all these cases, but it will not happen. So we have no patient cohorts, no standardized data. So it's impossible to really demonstrate efficacy of phage therapy. And we also confronted with another problem, which is a very old problem. And you could call it the Arrowsmith dilemma. I don't know how many of you know Arrowsmith. It's a book written by Sinclair Lewis in 1926, and he got a Pulitzer Prize for this book. And it was also turned into a movie in 1931. It came second in the Oscar nominations. So it was quite a good movie at that time. But it also showed that phage therapy then was known by people, even in the 20s. So, but this book, what is special in this book is that it's full of really interesting knowledge, because it was based upon the experience of the famous bacteriologist Paul de Kruijf, who knew about phage therapy and was very interested in phage therapy. Now, this book is about Martin Arrowsmith, a young medical doctor, who discovers a phage that destroys Yersinia pestis, of course, the bacterium which causes the bubonic plague. And he was confronted with a fundamental dilemma of medical research, and that is, should he pursue scientific precision through randomized controlled trials, or should he provide immediate relief to critical patients? So this is also our dilemma. So often when we present phage therapy at conferences, there are some people that are criticizing the fact that we are delivering phages without RCTs that are showing efficacy. And they feel that by doing this, we take away the incentive from the medical doctors to perform RCTs. Now, I will illustrate this dilemma with some excerpts from the movie, and I hope you will be able to hear this.
What is the experiment?
It is agreed that it will be of value to know what this stuff of yours is good for.
Why, yes, of course.
Well, if you could give injections to, say, only half your patients, and sternly deprive the other half of them, then you would know.
I certainly would, yes.
That is the experiment, Martin.
Well, I've done that sort of thing with cattle, but good Lord, Chief, these are human beings.
So you're just a country doctor after all.
Country doctor be blowed. I've got the bowels of compassion in me. These people are going to be dying and begging me for help.
And it's up to you to stand up and take it on the chin.
So Martin Arrowsmith, he wants to use his phages to cure desperate patients that are dying from the plague. But his boss, he wants him to perform randomized control trials and not to give the phages to dying patients. So after some discussion, Martin Arrowsmith agrees to perform randomized control trials, and he travels to a Caribbean island. It is not specified in the book which island, but it's an island where every year they have seasonal outbreaks of the plague. And his wife travels with him. And so he gives phages prophylactically to half of the population of this island, and he gives physiological water to the other half. Unfortunately for him, his wife did not get phages and dies.
I loved you, Lee. You knew that, didn't you? Didn't you know I couldn't love anyone else? Did you know that, Lee? I'll give them all the serum. Shoot them full of it. Come on, open the doors. Let them all in. Open the doors.
So after his wife has died from the plague, he decides to give the phages to every person on the island and to save the patients that were dying. So this illustrates the difference between being at your desk or at a regulator or at a company at your desk and wanting randomized controlled trials and being on the field and being confronted with patients dying. It's not so easy to say, no, you will not get phages. We will put you in one of two groups and you will get either phage or not. So of course it's because it's a natural tendency of clinicians to help patients. So that's why we say that we should do both. So we should try to already help dying patients, but also try to perform these RCTs. So what are our future plans? Well, to overcome these problems of standardization and having no standard data, we established a phage therapy committee, a national committee, for the coordination and standardization of phage therapy products and protocols. We had one meeting, and there are people from the regulator, from the certifying labs, from the hospitals, and we will have a second meeting in September. Of course, we need to perform phage therapy RCTs, but for that we need funding. And we have submitted some proposals and we hope to get some funding. Of course, we would like to have a centralized database for phage therapy cases. And if possible, it should be mandatory. Now, we started doing this. The analysis of the 66 cases was possible because these cases have been put into our database at the military hospital. And we will further develop this database in collaboration with the Belgian Institute for Health. But there's also an international database that is being set up at the Barbust University Hospital. And we decided to integrate our database into that initiative. And this initiative is really in progress of development. A lot of the development will be about data transfer and consortium agreements and privacy protection and stuff like that. But already we have quite some collaborators from Europe, the US, Australia, Israel. And anyone who is interested of collaborating should contact Shauna McCallan, who is the principal investigator of this project. Then, of course, we need to produce more phages, and this in collaboration with industry, because my bosses say that I cannot have more money to produce more phages. So this is not your task, they say. It's a task of industry to produce medicinal products. So we need to find collaborations with companies. We are developing in collaboration with Fissile Bioscience. And also we would like to develop cell-free phage production. So why is that? It's because this personalized approach has some major issues, because it's nice to match phage and bacteria, but as you can see here, this is what happened to us in Brussels. You can see all those arrows, and these arrows are phages and bacteria coming to Brussels for matching, are phages going to other places for treatment, or are patients coming to Brussels for treatment. That's the result of personalized approaches. And we have some, what we call phage therapy dream, how to solve this problem. It's kind of complex, but it has been published. So if you want to know the details, it would be a community-based approach where from the community, phages and matching bacteria would be provided. These would be sequenced, and then artificial intelligence would predict the genome of the phage that would target a certain bacterium. And this phage would be produced on site, so in the hospitals or even at home if you would want, by a device which we call phage beam. And these phages would be synthetic phage. And of course, this entire system would be managed by what is called distributed ledger technology, and would be fueled by what we have called a phage coin. This is all future, of course, but we started developing some parts of this scheme, and we got some funding from the Military Academy to do a proof of principle test on this production of synthetic phages. So from matching phage and bacteria that we would sequence, and then we would put them into an algorithm, artificial intelligence algorithm, that would then predict the sequence of the good phage. And then this phage would be produced in cell-free systems, transcription translation systems, to produce a synthetic phage. And then it's time to thank my colleagues, my phage colleagues in Brussels, and also to thank you. Yahoo! And you can contact me for more information if you want.
Thank you very much. Okay, here we are. Thank you very much, Jean-Paul.
You're welcome.
That was really great, and of course, it demonstrates both the challenges, but also the potential.